Welcome all, and thank you for joining today's AWR webinar. My name is Trent Weir, and I'll be your host today. In today's session, we'll take a look at rootstocks with a webinar titled Developing Pest-Resistant and Climate-Resilient Rootstocks. In the spirit of reconciliation, the AWR acknowledges the traditional custodians of country throughout Australia and their connections to land, sea, and community. We pay our respects to their elders past and present and extend that respect to all Aboriginal and Torres Strait Islander peoples today. I'd also like to acknowledge Wine Australia for providing funding and support for webinars via the AWRI's Extension and Communication Project. Before we jump in and make a start, a couple of quick reminders to anyone who is new to AWR webinars. If you'd like to provide a comment or ask a question, please click on the Q&A button on the Zoom toolbar. Type in your question and click to send it through. We'll be holding the Q&A session at the end of this presentation, but feel free to send in your questions at any stage. A reminder also that this session is being recorded and a link sent through to you later via the AWRI's YouTube channel. Anyone who has just joined, welcome. Today's webinar topic is developing pest-resistant and climate-resilient rootstocks, and it is with great pleasure to welcome Dr. Harley Smith from the CSIRO. Dr. Harley Smith is the research team leader for the Perennial Crop Improvement Team in the Trait Innovation Group at CSIRO Agriculture and Food. His main research interests are focused on the development of next-generation grapevine rootstocks and improving crop production in horticulture crops. Harley has established a diverse research team with extensive capability in perennial crop genetics, breeding, physiology, pest-plant interactions, developmental biology, and data analytics. Together, these capabilities are being utilised to deliver new climate-resilient rootstocks with long-term resistance to phylloxera and root knot nematode to increase vineyard performance to the Australian wine grape industry. In addition, the team utilises these capabilities to address the underlying biological drivers of avocado irregular bearing to develop practical management tools aimed at stabilising production for international and domestic market development. So with that, Harley, if you're ready to make a start, I'll get you to share your screen and hand over to you.
All right. Thank you, Trent, for the introduction. So can you see my slides?
Yeah.
Right. All right. Thanks again for the introduction and the opportunity to present today at the AWRI webinar. As Trent pointed out, the focus of this presentation will be on developing durable pest-resistant and climate-resilient rootstocks for Australia conditions. This work is funded by Wine Australia and CSIRO. So the two major pests that impact Australian grape production is phylloxera, the #1 biosecurity threat, as well as root knot nematode. Both of these pests can feed and reproduce on domesticated grapevines and susceptible rootstocks, leading to a decline in vine performance and in many cases vine death. By grafting domesticated grapevines onto rootstocks derived from North American vitis species that harbor resistant elements or genes to phylloxera root knot nematode has then the capacity to transform these infested vineyards into vineyards that are productive, but also in the case of Australia, to also protect those vineyards from the introduction of these pests. So the two major industry challenges in Australia with regards to rootstocks is #1, there's still numerous vineyards planted on own-rooted vines, which are highly susceptible to phylloxera root knot nematode. And that becomes a particular problem if there gets an introduction, say, to phylloxera to a region that are planted on they're on own-rooted vines, as that could cause significant economic losses. The other is that commercial rootstocks lack long-term resistance, and this has become really apparent with the emergence of aggressive strains. So for example, with phylloxera, in both Europe and California, now recently in Australia, there's been the emergence of aggressive populations that have emerged that can effectively feed and reproduce on rootstocks with Vitis riparia parentage. And this here is just an image taken of a 101-14 rootstock showing these aggressive phylloxera adapted to feeding on these roots in California. And this then causes both the swelling and distortion of the root that likely impacts root function. In Australia, one of the more popular rootstocks used in production is 1103 Paulson. It's been characterized as having moderate to high resistance to root knot nematode. However, over the past few decades, there's been a number of populations that have emerged that can effectively feed and reproduce on the roots of this rootstock. So there's been population identified in McLaren Vale, the Barossa, the Riverland, and out along the Murray River in New South Wales and Victoria. In addition to these pests, the Australian grape and wine industry also faces challenges associated with climate change. So many of the wine grape growing regions, it's been predicted that these regions will suffer from drought, which will then reduce water availability for irrigation. There's also, with drought comes a decrease in water quality that can then impact soil salinity, and then also extreme heat events. And then, of course, in the off years, you can have flooding and cold temperatures at flowering that can impact production. So our goal with the CSIRO Rootstock Breeding Program is to develop durable pest-resistant rootstocks adapted to a changing climate in Australia. So one of the first questions we can ask is, how do we breed for durable or long-term resistance? So if we take a step back and look at traditional breeding programs, they've been very effective at deploying material containing a single resistant trait that can provide resistance in the short term. However, if any selection pressure is placed on the pest or pathogens in the soil, those pests or pathogens can evolve and break that resistance. And a good example of this was in the Central Valley of California when they did widespread plantings of Freedom and Harmony. And those plantings placed selection pressure on the root knot nematodes in those soils, and there were many resistant breaking populations that emerged that broke resistance in Freedom and Harmony. And what's a problem here, in addition to this, is that any of the rootstocks that are related to Freedom and Harmony, also those nematodes can also break that resistance. So for example, Ramsey, those root knot nematode resistant populations can also break resistance in Ramsey. So the way that we breed for long-term resistance is we take two resistant traits that act in a non-redundant manner, and we stack them or combine them into an individual rootstock. And then when that's deployed to the field, the idea here is that it will be very difficult for a pest or pathogen to evolve and break two resistant, non-redundant functioning resistant loci or traits simultaneously. And that's how we breed for long-term resistance. So the next question we can ask, well, how do you stack and select for multiple resistant traits in a rootstock? And the approach that we're taking is DNA marker-assisted selection, where we use DNA markers linked to the resistant traits and then use those as proxies to select for rootstocks that have these Phloroxera or root knot nematode resistant traits. So these act as a way to indirectly select for resistance. And the advantage here is that we can screen through hundreds of thousands of plants at the seedling stage without having to go through laborious glasshouse screening methods, where our ability to screen seedlings, the number of seedlings is highly reduced. The other important thing here is that these markers are specific, so we can detect rootstocks that have multiple resistant traits. So how do we identify these markers? Is we use genetic mapping. So we genetically map these loci and then look for polymorphisms that we can then create DNA markers. So we can ask, what are the ideal traits to target for marker-assisted selection? So the ideal traits are going to be ones that are controlled by a single gene or a few genes. And I'll show you that in the case for root knot nematode and Phloroxera. Traits like drought tolerance are highly complex and likely controlled by hundreds of thousands of genes in an additive manner. So taking this approach to study a complex trait is really not feasible. So the way that we're approaching things like drought situations where there'll be limited amount of water for irrigation is to set up field trials. And I'll talk about that later on in my presentation. So now I'll switch gears and talk about Phloroxera. So Phloroxera is subdivided into different biotypes, and I'm only going to touch on 3 of the biotypes. So biotype A Phloroxera are populations and genotypes that are adapted to feeding on Vitis vinifera. Biotype B Phloroxera are populations and genotypes adapting on hybrids between Vitis vinifera and North American Vitis species. And biotype C are populations and genotypes that are adapted to feeding on material of North American Vitis species. So in Australia, Phloroxera is managed in part through quarantine measures. And this here is a map of the southeastern corner of Australia. And you can see here that these red boxes depict the quarantine infested zones where Phloroxera is known to exist. And then there's also Phloroxera exclusion zones and risk zones. So these quarantine measures have been pretty good about limiting the spread of Phloroxera, but they're not foolproof. So for example, in 2006, Phloroxera was introduced to the Yarra Valley, shown here, in 2006. And in 2019, a publication came out in Vine Health that estimated that this cost the industry up to $1 billion. So these quarantine measures are good, but on top of that, I think it's also important to utilize rootstocks that have Phloroxera resistance. So Vitis cinerea C2-50 is one of our primary breeding lines, and through collaboration with AgVic, we've been able to show that this provides resistance to the two biotype A superclones, G1 and G4. And then it's also recently been shown to be able to provide resistance to G38. And G38 is one of these new populations, or it's a population now that's been identified that can effectively feed and reproduce on 1 and 114, Schwartzman, and 3309C. So in 2018, we published an article where we mapped the second Phloroxera-resistant locus in grapevine in Vitis cinerea C2-50. This trait or locus mapped to the near the beginning of chromosome 14. And this mapping was based upon resistance to G1 and G4 biotype Phloroxera. We recently asked the question, does RDV2 provide resistance to G38? So we screened a population that was segregating for RDV2, and what we found that it did not provide resistance. So RDV2 didn't segregate with G38 resistance. And in fact, what we're finding is that there's a second locus that we're currently calling RDV38 in Vitis cinerea C2-50 that provides resistance to G38. And with RDV38, it would only provide resistance to G38, but not G1 and G4. And what this suggests is that Phloroxera resistance is complex and may require biotype-specific resistant genes to manage the different biotypes of Phloroxera, or maybe even genotypes. Okay, now switch gears and talk about root knot nematode. So in Australia, there's 3 major root knot nematode species that inhabit vineyards. So that includes Meloidognia javanica, Incanita, and Arenaria. So in 2018, we mapped the first root knot nematode resistant locus in Vitis cinerea C2-50, which mapped near the end of chromosome 18. In our screening, we maintain 3 populations of root knot nematodes, so a javanica population, an Arenaria population, and an Incanita population. And what we're able to show is that this MGR1 was able to provide resistance to all 3 populations. So this would suggest that MGR1 would provide broad spectrum resistance to the 3 species of root knot nematode. Currently, we're mapping MGR2 in Vitis champini. And one of the reasons why we're doing this is that work from Peter Cousins when he was at the USDA, work from his studies there suggested that the mechanism of root knot nematode resistance in Vitis cinerea is distinct from that of Vitis champini. So combining those 2 would allow for a durable resistance. And then we're also mapping a third root knot nematode resistant locus in Vitis candicans. Okay, so our group and other groups throughout the world have mapped both Phloroxera and root knot nematode resistant loci. And so what we're currently doing is using DNA markers linked to some of these Phloroxera and root knot nematode resistant loci to generate new rootstocks. So currently, We're not only using markers linked to RDV2 and RDV38, but also markers linked to RDV1, which was identified by a group in Geissenheim, Germany, which was the first phloroxera-resistant locus identified. And then we plan to stack in MGR1 and MGR2 into these rootstocks as well. Once these resistant loci are stacked, we can then go through vine multiplication, which will then allow us to establish nursery and field trials. Now, when we're doing our breeding, we're thinking about breeding for rootstocks in the future. And we know that from climate modeling that in Australia, it's expected that temperatures are going to rise with more hot days and fewer cooler days. And so we ask the question, is root knot nematode resistance stable at elevated temperatures? And so we developed both a glass house and an in vitro based screening system. And what I'll do is show you some of the results from the in vitro screening. So this is screening performed in vitro at 27 degrees Celsius with Chardonnay as our susceptible control and Freedom and Schwartzman, which have resistance to this M. javanica population. The control panels, A, J, and P, show the appearance and morphology of roots in the absence of this M. javanica nematode. But when we add it to Chardonnay, we get galls forming, and eventually those galls will split open, producing this callus mass. And shortly after that, we'll start to visualize new nematodes or new infectious nematodes in the media, indicating that the nematode can reproduce. Now, in contrast, when we look at Freedom and Schwartzman, when we add nematodes to the media, we observe these localized necrotic responses occurring in the tips of the root, and it's likely these function to trap the nematode and prevent it from establishing a feeding site so it can reproduce. So we get complete resistance at 27. When we perform the screening at 32 degrees Celsius with these rootstocks, we see a different story. So the control panels A and E are the images, the appearance of roots, both the and morphology of roots at this temperature. And then the other panels are what happens when we add this M. javanica nematode. What we observe in B and F is we do get some resistant responses, but also we get susceptible responses. So we'll get gall formation and also these callus protrusions. And shortly after those are produced, we'll start to see infectious nematodes in the media that can then go and infect new roots. So it looks like at 32 degrees Celsius, there's a breakdown in nematode resistance in Freedom and Schwartzman. So what we think is going on is that could either be due to the resistant gene, but probably more likely it's going to be due to genetic modifiers that influence the stability of this resistant locus or loci when the temperature is raised. And because Vitis vinifera, or because Vitis species are highly heterozygous, and we don't know the nature of these genetic modifiers, what we're doing is that after we select for rootstocks that have these phloroxera and root knot nematode resistant loci, we will put them through our high temperature screening assays, both in vitro and glasshouse, to then select for individuals that maintain a high level of resistance at 32 degrees Celsius. And then those ones we'll put through vine multiplication and put through our nursery and field trial evaluations. Now, in the future, when these rootstocks get to that stage, one of the important trials we need to do are nursery trials. And here, what we want to evaluate is the ability of these rootstocks to form and develop adventitious roots and how well they graft with some of the top producing scion varieties. And one of the important things we need to be doing in our evaluations is ensuring that they're performed in line with industry practices. And we feel like these nursery trials are really important because if any of these rootstocks don't form roots or don't graft well, there's no way that a nursery person's going to want to work with it. So these nursery trials we think are going to be key for adoption and uptake. In the future, we also will perform field trials where we'll evaluate the impact of rootstocks on yield, vigor, berry quality, berry composition, and wine quality. And we plan to do these in Sunraysia in a hot climate, in the Barossa in a warm climate, And it would be really nice to be able to set up a trial in a Phlox infested zone to evaluate their performance under Phlox infested soils. Now, to address the changing climate, of course, as I mentioned earlier, drought is expected to occur in many of the wine grape growing regions, which will reduce irrigation levels. And so we'll want to set up trials to select for rootstocks that perform well under water deficit conditions. Salinity is another important issue to address, and work from Jake Dunleavy and Mandy Walker's team had identified DNA markers linked to salinity tolerance genes, and so we're currently using that in our breeding, and we know that our parental lines that we're working with contain these salinity tolerance genes. And then lastly, by performing our trials in Sunraysia at the CSIRO Imperial Farm, we're hoping to capture the impact that extreme heat events can have on these can have on these rootstocks. Okay, so that's all I have to say about our breeding. I'd like to thank the people that have contributed to this work and the people that are currently contributing to this work, as shown here in these panels, and also our collaborator in AgVic for their fluoroxyl screening capability, which is really essential for mapping these fluoroxyl-resistant loci. And lastly, I'd like to thank Wine Australia and CSIRO, and thank you for your attention. Happy to take questions.
Thank you very much, Harley. There has been one question come through. Someone's just wondering about when developing climate resilient rootstocks, is there any that you have worked with or any scope for work in the future, looking into ones that delay ripening?
Yeah, that's something we can look at as the impact of the rootstocks and phenology. Definitely. So that's something we can monitor, and we plan to monitor both phenology. I should have mentioned that, but yeah.
Yeah. So is that something that's monitored in your work that you're doing with these ones, or is that more looking at future projects for that?
Yeah, future projects. So we haven't released any these, none of these rootstocks have been developed. So this is just what
Yeah.
I'm just presented as our pathway. But once we get down that line, that's something we like to do. And I think well, our plan is to try to adapt some of the digital imaging technologies that Everard Edwards developed, so we can capture more time points throughout the season in a non-destructive manner. So to
Yeah.
capture effects on phenology, vigor, and so on, and berry quality and composition.
Another one that we've had come through is wondering what sort of timescales are involved to work through this process that you're currently undertaking.
Very good question. So our time scales is we hope to have the first set of next generation rootstocks ready to be released by 2032.
Yeah. Yeah. And when was it that this work started?
We started the genetic mapping back in 2015.
Okay, great. Perfect. All right. Well, thanks very much for taking us through your work. I'm sure everyone who attended today got a lot out of this session. I'd like to thank you, the audience, for logging in and taking a part. I'd like to remind you that, as always, you'll receive a link to view the recording of this presentation, which will be available on the AWI's YouTube channel. The next webinar will be held on next Thursday, the 12th of June, with a webinar titled Vineyard Biodiversity, supporting the sustainable management of scale insects and other pests. So look out for this advertisement coming soon. If you'd like to register for this session, please visit the AWI website. Thank you again, and thank you, Harley. And we look forward to seeing everyone at the next AWI webinar.
Thanks again.